The Seeking Eye Life Exploration podcast is and will always be free to access. But if you would like to support my work, please consider joining my Patreon or signing up on YouTube as a channel member. By doing so, you will receive exclusive benefits, including early access to future podcast episodes, access to hosted discussion groups, and access to non-podcast supplementary videos. So click the join button underneath this video to join as a YouTube channel member, or find the link to my Patreon page in the description. Thank you.
If you saw, there was a graph in my book which I showed, and that was John Sutherland, who's one of the leading, well, I don't know if he still is, but he definitely was at the time I wrote the book about 2018, one of the leading lights in origin of life research. And his conclusion was, after all these years, there really hasn't been that much progress since then. That we haven't really gone beyond even closely getting some sort of idea of what might have happened at the beginning. And so you've got almost like an exponential climb in terms of what's really conceptual ideas after that. You don't need to be a chemist really to get your head around the conceptual ideas, because they're very much just, obviously inconceivable how you would solve them. And that's really what most of my book focuses on. But yeah, Farina and Tor spent a lot of time arguing about chemistry, really. And Farina was frankly a bit dishonest about the way he represented things. The chemistry, people have shown in various papers that under certain conditions, you can get a reaction to happen, and you can get, some sort of nucleotide or an amino acid or whatever to form, and you might get the right chirality if you shove it on the right clay and so on. But the idea that you would get large amounts of these materials in sufficient diversity, because these molecules, you need all four, nucleic acids, and you need, probably at least 10 to 14 amino acids to really...
insufficient purity as well.
You would have no purity. To get purity in an uncontrolled environment is just beyond, chemical reality. I've worked in a lab. I did my PhD was in organic chemistry, and I was working with nucleosides and nucleotides. I was working with amino acids. I was specifically working on targets that disrupted DNA synthesis in viral cells. And so, you are ultimately... I'm very familiar with the biochemistry and chemistry of these compounds, and if there's even the smallest amount of, if there's anything there, any impurity, more than 2 or 3%, then the reaction just turns to a mess, and you end up, it just becomes a complete mess. And then you do a reaction under the most controlled conditions. You have to purify it, you have to separate it from various things. There'll be there'll be side products and everything else. It's fantasy, and they know, to be honest, they know. But it's a bit of a distraction, because they can say that, oh, look, we've done this and we've shown this can work. Well, they haven't really. They might have shown once that I'm repeating what Tua said, but it's what I say in my book, which I wrote long before I'd seen that. But Sutherland, really, the point that he makes is that even if all of that's true, even if everything that everything that Farina says and everything that the others in the field say is true, they still haven't got beyond the very earliest sort of things, how to get the starting materials and how to sort of get some reactions going that might produce some of these larger macro molecules, the polypeptides, the polysaccharides, and so on. So, you mentioned to me in the last video and also online in communications about that debate. And, this is quite typical of what I've experienced. Farina outright told a mistruth. Don't know if he was deliberately lying or whether he just didn't know the data. But there never has been produced in a lab or anywhere a fully self-replicating piece of RNA. And it's very important. It's kind of a conceptual point that you need to reach, because you need to have one molecule that can pass on all its genetic material and at the same time replicate that genetic material. Either that, or you have a complete system that's able to do that. There's only those two options, and the chances of having a complete system in an environment where there's no cell wall are basically impossible, because they just float off. So you have to have either. So this whole idea of generating a self-replicating RNA molecule is important, because if they say, oh, you can get one of these, and maybe the RNA world, has some validity, which it doesn't. And I'll go on to that in a minute for sure. But the reason why is, so imagine, I mean, this is my take on it, and I may be wrong, but I don't think so. James Tour, in that debate, he said, the best they've got is about 10% of the molecule being replicated, right? And I'm not going to check it up, because he's probably more up to date on the data than I am. But so you imagine you've got a snake, right? And snake sees something waggling in front of him. The snake's hungry. So the snake reaches out and grabs it, and then starts eating this thing, and goes along and along. Suddenly realizes it's eating itself. Now, how far could the snake get, right? It can't eat its own head. And that is the key issue with a self-replicating RNA molecule. The active site can't really replicate itself, because it's the active site. But conceptually, they don't really talk about that. I stopped reading these papers a long time ago, because I usually found the headlines never matched what was basically endless amounts of scientific word salad that, even to someone who's got knowledge didn't really understand. So, I think what you've read to a large extent sort of goes into that a little bit. But yeah, anyway, I'm just talking.
No, that's the idea. So you know a lot more about this than me. The only thing I had seen really on Origin of Life after talking to Dave was his debate with James Tour, and most of it went completely over my head, of course, as it would do.
That's what they rely on. They rely on that, and it's blinding people with science. It's a real term, and it's a real thing. And I mean, I read a book by Nick Lane, who's of the same ilk, and it was, I think it's called Life Ascending, Nature's 10 Greatest Inventions, and he talks about the origins of life, and particularly the change in the code. At some point, you'd have to go from probably a doublet DNA system, which is where you've got two nucleosides, and I know this is going to be above people's heads, but you've got basically two letters, which are nucleosides, and they are letters, coding for an amino acid, to three nucleosides coding for an amino acid. It's conceptually impossible. It might seem like, oh, that doesn't seem too difficult, but it's conceptually impossible. And I'd like to talk about one example of that. It should be even easier than that. But you'd have to change the whole system. It's like going from Microsoft to iOS in one cell cycle. It's just, and what he wrote, he wrote 2 pages on this in his book. I looked at it and I thought it was just not, it didn't make any sense. It was just gobbledygook. And I mean, I do understand this. I know what this is all about. And it was just basically a scientific word salad. And he was relying on, people who read it, the journalists who reviewed it slavishly, to, say how wonderful it was. And it wasn't. It was just garbage. So ultimately, there is a lot of that, and a lot of relying on people not understanding and just choosing their side. But there are a lot of people with scientific expertise much greater than me. I mean, I've not been, been doing chemistry for a very long time, but I have been in fields that, work on disrupting DNA in viruses or in other processes. And so I'm familiar with the biochemistry, and I think the biochemistry is perhaps more relevant to the conceptual issues with the origins of life, because it gives you a slightly zoomed out picture. Biology is almost too far zoomed out. So when you talk about evolutionary biology, you zoomed out too far. When you go down to, independent organic reactions, peptide bond formation, chirality of, the sugars and so on. Ultimately, it's going to go over people's heads, but it's also, you're getting too close to the problem. You're not seeing the bigger picture. Whereas biochemistry, you see the bigger picture. And then some people go down into quantum level, which no one ever understands. But yeah, no, it was painful watching that debate. And I've had my own experiences where I've dealt with people of a similar ilk whose only answer to the scientific questions I have is to hurl abuse and insults. And it just says everything about the arguments they have. They don't have an argument. And something that Farina did, which, again, it looks really convincing, is to talk, this paper says this. And I was like, why doesn't he say what the paper says? He just shows a headline. And eventually Tour said, you're not explaining what the paper says. Either he can't or he doesn't want to, because often the headline of the paper is very different. Because I've worked in writing papers in science for a long time, usually on behalf of others or with others. And the headline is to grab attention, as always. And often the reality is somewhat different. So, yeah.
And that's one thing I was going to bring up, because Dave certainly seemed to have done, his homework on this. I don't know his background in Origin of Life. I think he said he's not particularly, that's not like a focus interest of his.
No.
But he did certainly seem to know the papers and know the literature. As you say, it was just argued against whether those papers were really showing what he claimed they were. And I have to take people's word for these things, because I don't know.
Well, I haven't read them either. And this is the thing, there are thousands of papers written on this, and neither of them read all of them. At one point, Farina was saying, "Oh, well, you haven't read these papers. You're an expert on the field." And Tor made the point that ultimately you can't read all these papers, and a lot of them are garbage. So why would you read them? And then Farina got called out, and this is the thing. It's like there's endless papers out there. If there had been a real breakthrough, you'd know about it, and it would be something that if you Googled it or you went on. So, for instance, if you go on ChatGPT, which reasonable, does a reasonable job of trawling the latest sort of thinking. The problem's far from solved. This hasn't made great progress. Just not true, because there hasn't been great progress made. They get at the very best. If you said all those papers were right and they said what Farina said, then you've got to the starting line, right? You've got the means by which you can start to create some of these large macromolecules that do the actual work of life. But you don't overcome the huge conceptual problems, which ultimately mean it doesn't matter if you can get the chemistry right. It doesn't matter if you can sort out chirality. None of these things matter, because it's about the conceptual barriers that are so vast that no one can actually conceive of how you'd overcome them.
So can we maybe start, because as I say, I've made it about a third through your book, which is kind of the introductory things. And again, I'm coming at this from a completely no knowledge of any kind of organic chemistry at all. So can we maybe start from the very beginning and talk about some of the key fundamentals that you mentioned, such as chirality and various other starting point issues?
Yeah. So, chirality is, it's one of the key issues. So if you look at the sugars that make part of DNA, for some reason it's gone out of my head. It's furious. I can't remember the name of the ring, but it's basically a pentagon with an oxygen in it, and then all of the other. So chirality. It's really hard to explain if you don't.
It's easier to explain with graphics, isn't it?
Yeah, it is. And obviously this is a podcast, so trying to make people appreciate the sort of, you've got 4 atoms that are equally spaced in a 3-dimensional structure around a central atom. So you've got 4 bonds, and you can have the same groups on each bond, but you change the way that they are. Let's say you've got 1 bond is 1, you've got 2, 3, and 4. If you change the position on bond 2, molecule or atom on bond 2, and you swap them with 3, it actually completely changes the properties, even though you've got exactly the same things attached. So, for instance, the thalidomide, you may not be familiar with this. It was something that happened in the 60s, and it was a drug that was used to try and treat morning sickness. And it was basically they made it something which had the same components around one carbon atom. Unfortunately, one caused huge birth defects. The other worked fine and didn't cause any birth defects. So, and that's the kind of example. So when you look at nature and you look specifically at cellular structures, all of the every single molecule in the amino acid, let's say, has to be oh my goodness, my brain is just so hot today. And then it's L for the nucleosides in DNA. So ultimately, you have that's L for the amino acids. Sorry, it's L in proteins. It must be D, and I always get it muddled up. So honestly, I don't bother with this so much anymore because I just talk about NDEs most of my time. But it doesn't matter because either way, you have to have the same chirality all the way through. If one of those bonds, and like, so for example, you've got a chain of maybe 1,000 amino acids for a protein, just one of those bonds is wrong, then it doesn't work. And normally when you bring molecules together outside of specifically controlled conditions in a laboratory, you'll get both in equal amounts. So you either use an enzyme, which is a natural product, natural it's a protein that does the work for you if you can, or you use a specific catalyst that will make sure you just form the one bond, or if there's sufficiently different properties, you can maybe filter it or separate it that way. Nature, in a random environment, you wouldn't have that, and you just get a mixture. And if you just have one of those bonds, so as a chain forms, if you have one of those bonds not forming the right orientation, and it all falls apart. And that's both for DNA or RNA, and the same for proteins. So ultimately, chirality is a huge issue. They've claimed to come up with solutions involving certain types of clay and whatnot. You can go, yeah, okay. But in reality, it's absurd. It's just absurd to conceive that these things would happen like that in that way. But you can just give it away because it's such a small part of the problem, actually. And it's not just that. There's so many for some of these reactions. When you have a reaction, you could have but they're like you've got several. I'm just going to have to Google this, driving me nuts.
So while you're doing that, just to kind of clarify the idea on the kind of the thalidomide example. So effectively, thalidomide was this one, is it compound, I suppose would be the term?
Ribose. Sorry. Oh my goodness, yeah. It's hot.
So thalidomide would be this one, I suppose you'd call it a compound with various bonds to other molecules. And the molecules were all exactly the same on both types, except that the positions of two of the molecules were swapped. And despite the molecules being exactly the same, but in a different order, they had completely different effects.
Yeah. So if you imagine you've got a central atom and you have four things coming out of it equally spaced. So when you put it on the ground, it would look like, well, if you could turn into like the Eiffel Tower or something like that, that's got four at the bottom. But it's not the same molecule, even though the four things are exactly the same, come from that central atom. You just swap two of the locations. It changes the properties completely. I wish you could edit out the Furos. I can't remember where that came from in my head. That's why it's called RNA. Anyway, so ultimately, and also you've got all these different active sites on some of these molecules to react with. So like the ribose ring has got these 4 or 5 reactive sites where you do a reaction and it could react to one of them. Now there'll be a paper somewhere that says, but if you use these conditions in this way, then you'll get that. But so those conditions had to exist as well as the other conditions, as well as the other miraculous, and it just starts to become absurd. But they'll say all of this, and they'll say all these things. This worked here and that worked there. Trying to bring it together doesn't make any sense, and but it's not the biggest problem. Not by any way, any chalk of the imagination. It's not the biggest problem. They've tried to solve their problem in the RNA world, but the RNA world is a giant red herring, and it's just quite pointless and doesn't actually achieve anything. But that's conceptual. The RNA world is a conceptual idea. But even to get to the RNA world is pretty much, they can't figure out how you get to the RNA world, really.
The RNA world is?
Right. So you've got DNA, which is deoxyribonucleic acid, and you've got RNA, which is ribonucleic acid. And basically what it is, you've got this, like I said, it's a pentagon. There's an oxygen at the top, and then you've got 4 carbon atoms forming the rest of the pentagon. Out of one bit, you've got the nucleic acid bit, and then the other bit, you've got the oxygen, carbon and oxygen atom. And then those two are the same in DNA and RNA. The RNA has got 2 oxygen atoms coming out of the bottom of this ring, oriented in the same way, whereas DNA has just got 1, so it's deoxyribose or deoxyribonucleic acid. It's got 1 oxygen atom missing, so it's deoxygenated. Now they have, otherwise they're the same, identical as molecules, and then they join together they can join together in the same way via a phosphate linkage, and they join at it's called the 3' and the 5' oxygens, and they can both form polymers. So you can get chains of RNA, and RNA appears in cells. It's vital, in fact, to cellular replication, and it's in the ribosome, which is found inside the cell. And what happens during the replication of a strand of DNA, mRNA will form along it and be sent into the cytoplasm, where it'll then go to the this ribosome, and then what happens is tRNA is brought into the ribosome already attached to the appropriate amino acid. Translation doesn't actually occur in the ribosome, although biology textbooks might say that. It doesn't. Translation occurs in the cytoplasm, is mediated by enzymes. And this is just like, people going like, blah, 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 blah. It is what it is. For those who initiate, they're not talking about. So there's amino acyl RNA synthetase, which basically brings together the right tRNA, the code, which is mobile form of a codon, with the amino acid. So basically, though, you've got these RNA molecules that are involved in translation. And you can actually build RNA molecules that act as enzymes. They're like proteins, but they do really special work. And everything, in your body is so many of your processes are mediated by enzymes, and it's always proteins. There's a couple of ribozymes, but mostly proteins. The fact is that these machines are phenomenally accurate and incredible. They're micro machines that do ridiculously amazing work at super fast pace in each cell. And you can make ribozymes, which are ribonucleic acid equivalents that are similar. So the theory is that you could have a world where RNA, which has similar properties to DNA in that you could have a code. You need DNA to form the code. This is why you need to read the book. Honestly, it's just like you need to go through this and give yourself time to actually absorb it. And it's just like the RNA world is basically, and I go into a lot of detail in my book. I didn't really want to talk about it actually in this because it is too much. You need hours to talk about why the RNA world is a load of horse manure. But the reason why it came up as a theory is because you can make code using RNA just like you would with DNA, but 2, it can also act as an enzyme. So you've got this world in which you've got this material that does both jobs, storing a code and acting as an enzyme. And if you stayed awake for that, very well done. But ultimately, the problem is that there's no clear routine. It's not particularly stable RNA. At some point, you're going to get a cell wall. there's no, and we'll come on to that, 'cause I think that's a simpler concept. I wanted to try and just talk about simple stuff. So the cell, that's why I say that reading the book doesn't, there's a lot to it. I had a doctor friend of mine. He took 4 or 5 weeks to read it because it's not straightforward. So, but so the idea is that you can't. There's no way into the RNA world that seems to work, because if you haven't got a cell wall, cell membrane rather, then you can't keep these materials together. So you have to have a self-replicating RNA, and then you have to have lots and lots and lots and lots and lots of pure raw materials, the nucleosides. And then, but the biggest thing is transitioning one from an RNA world to an RNA protein world, and then 2 to an RNA code system to a DNA code system. And it's just, again, it's a few pages in my books to explain why that is inconceivable, how you could do that. And the only answers I've ever read are scientific word salads that don't actually make any sense.
And I know you do have a chapter dedicated to so-called big numbers, where I'd imagine, I haven't got there yet, but where I imagine you go into things like, probability, because most people from a lay point of view would think, well, as unlikely as these occurrences seem to be, we're talking about a huge planet here, with multi-trillions of these molecules all interacting together. It's bound to happen at some point. That's what most people would imagine.
Okay, so so just as it's impossible to imagine how big the universe is and how much time there is over times, it's also impossible to imagine or understand how big these numbers are. So one that's often quoted, and they try and sort of argue against this, but it's an example that has been mathematically looked at, and it was calculated by Seth Lloyd, who was an MIT postgraduate or postdoc, and he basically did a calculation as to the likelihood of a functional protein forming 150 amino acid long functional.
I think you mentioned this example earlier in the book, didn't you?
Yeah, and so it's just quite a small, probably about the smallest enzyme you could get. You might be able to get something slightly smaller than that, but you've got huge variability into how that would form and everything else in the reaction sites and so on. And he came up with a number that's 1 times 10 to the 164. So as to the likelihood of that happening. So that's 10 times 10, 164 times. You go, well, the Earth's a big place covered in these molecules. Well, first of all, it was never covered in these molecules, ever, 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 ever. There would be evidence if it had been covered in these molecules. There might have been a puddle somewhere that had a few of these molecules in it, if you're lucky, really lucky, right? So that's the first thing. But he sort of comes out with some other numbers. So if you took all of the atoms in the universe, it's 1 times 10 to the 80. So there's not near 1 times 10 to the 164. If you took all the particles in the universe, I think it only just goes to 1 times 10 to the 90. If you then took all of the jiffies since the beginning of the creation of the universe, and the jiffy is the smallest amount of units of time. It's like a measurably small amount of time. It's almost an immeasurable amount of time. And then you included that, and then you allowed for, I think, the entire planet Earth, like you described, to be covered in amino acids, and they got together every jiffy in 150 now. You would still have a 1 times 10 to the 44 chance of getting one, which is, it's not possible. It's not reality. And, when you look at what number is impossible, then, this is impossible. And that's just one protein. So to translate DNA, I think it's about 80 proteins that are needed. In each cell, there are hundreds, if not thousands of proteins. And for a cell to function, you need all these working. If one of them goes wrong, you start getting diseases or stuff like that. So, it's just, it is not realistic to think that these things would come into being by random natural processes. And at some point, by one route or another, a protein had to come into existence. To do that, ultimately, it's the same mass whether you do it in bits, and bits wouldn't work. Smaller polypeptides, they can have functions, but they're not able to do the sort of complicated stuff which would need to be done for all these processes to get going. So, you talk about those numbers, and they are ridiculously big. And it doesn't matter how much material you've got, and even if you have all the time, the whole of the universe working towards this, it still wouldn't happen. That's how big the numbers are. And that's just what it is. So, wow.
So, yeah, I mean, as again, someone who's never really looked at this, my immediate kind of thought was you hear about the primordial soup and all that, and you imagine this barren landscape of Earth, early Earth, covered in lava and molten rock, which is just exactly the same thing in a different way. And you imagine all these pools covering the entire Earth's surface, filled with these amino acids and these organic chemicals. So, and you mentioned that you'd be lucky to find maybe 1 or 2 ponds of that cut here or there. So, what are the ideas of where these could have come from, if not spawned?
Well, that's a very good question. It's a very good question. I mean, it's been shown that you can generate amino acids and some of the components of nucleotides with those earlier materials, and there's bits that are sometimes found on meteorite. Whether they've been contaminated or not is another question. But it's likely that there was some, and not just maybe one puddle. There would be puddles or whatever with these materials in, but they would not be pure, and they would be in small amounts. And this is the point, that even if the whole Earth was covered in an ocean of these things, all perfectly pure, and the reactions occurred in a perfect way, it still wouldn't happen. That's the conceptual issue. It still wouldn't happen. But you haven't got that. You've got a messy environment. You've got meteors hitting the Earth. Well, I think they changed it recently that it's maybe possible that life could have formed a few 100 million years earlier than Luca. And Luca's a really important thing.
That's the last universal common ancestor?
Correct. So life divided into 3 groups. And that's happened at around, they reckon, either they moved the date of that, or I think they kept that date. And it was like 3.7, 3.8 billion years ago, something like that. And up until recently, they said, well, life really couldn't have, been sustained or appeared even much before that, maybe 100 million years. And I think they've given it another couple of 100 million years, but it's just not enough. Luca is essentially, it's the same it's very similar to the cells in your body. It uses the same protein DNA system that we use, which is insane. That's never evolved, really. It's never changed. There's a few examples of molecules that have got a different amino acid or one less amino acid or something like that. It's interesting thinking about why that might be the case, but there was I don't believe there was evolution involved in that. It's de-evolution, if anything. But where these materials come from, how this happened, because science really can't explain it. It cannot explain it. And if sure, you might be able to get, maybe a 10 or 15 amino acid chain that sort of, it's the idea that you're going to get these huge chains of amino acids forming perfect proteins that are going to then do a job on what? There's nothing else there. And then you've got, okay, at the same time, RNA forms, and that's difficult as well. Just as difficult. Actually, RNA is not very stable, and the conditions aren't brilliant for any stability of any chemicals, let alone an RNA molecule. Oh, here they are. It's just the whole thing's batty.
And even from trying to conceptualize, you say what 1 in 1 to the 10, was it 168 or something, and that's for 150 amino acid protein. So add that consecutively onto all the other proteins that need to be developed with longer chains of amino acids, and you're getting into numbers that, as you say, are just...
No, but once the 164 is insane. You don't need to add any more. You really just don't need to add any more. That is insane. It's just beyond, it's a number to you. You don't understand I don't really understand. I can't really conceive what it is, but I can look at the maths and then compare it to the maths of known things. It's beyond the realm of possibility. It's not going to happen. So I think that's it's an important point to understand the numbers, and it's a real point, and it can't be dodged, and they try and dodge it. But the truth is that at some point a protein has to come into existence. At some point, many proteins have to come into existence. Even if you had an RNA world, and in this RNA world, there was a machine, which suddenly started generating proteins, and there was a supply of amino acids lying around. And it's still beyond the realms of statistical reality for one of these machines to make a protein, and it happens to make a protein that's useful for a process, which is it's the whole thing is nuts. It's actually nuts.
Practically impossible for.
Yeah, it is impossible. It's impossible by natural means. It's impossible by random natural processes. And if you start to link in what we spoke about last time about how actually the universe around us isn't real, then you can start to maybe get a picture of why it's like this. And science speaks to the lack of reality of the universe through quantum mechanics and so on. And I don't fully understand that, but it's a distraction, for want of expression. So, but I think the conceptual thing. So I did send over the numbers thing. We sort of covered it. It's just statistically insane to believe that multiple macromolecules could ever form that had any purpose other than forming a guff at the bottom of a pond and never coming to anything. I think the second thing I sent over was the cell wall, which is more of a conceptual thing. I think is easier to explain in a way that's understandable, right? And to see why it's a chicken and egg situation, right? Or an eggshell and yolk situation, for want of a better expression. And it's one of these situations, if you don't have one, you can't have the other. So if you imagine you've got this nascent RNA world or another nucleic acid world with a different sugar on it, whatever you do, you've got some world that's sort of starting to do something. This little group of molecules that somehow formed against all the odds, and they started doing their stuff. But they're in a soup, right? If they're in a soup, they just drift off. They'll never work together. Or you have one molecule that's able, super molecule that's able to do everything, and we've already established that conceptually an RNA a fully self-replicating RNA molecule doesn't really make sense. It's the snake eating itself. We can't eat the head because of you've got one active site and you've got a chain. It can't work on itself. And anyway, so let's say you've got something like this. You have to stop it from floating off and going in different directions, right? You have to bring it together, and eventually there is, the only reason that cells work and do their thing is because you have a cell membrane, which is a highly specialized membrane. In our bodies, the cell membrane is coded for in DNA. The actual membrane isn't coded for in DNA. It consists of these sort of fatty molecules that I deal with this all the time. It's the heat. My memory's getting a bit ropey as well. But basically, it's got proteins in it. You've got, and it's all how these things are assembled is through proteins themselves. So the DNA codes for a lot of proteins and timings and everything else. The bits are assembled, the molecules are assembled in the cytoplasm, and then you've got a budding cell wall, and it assembles it, and off you go. So a membrane. So that's how it happens in us. So you have to get to there, right? And the way they sort of say is, well, first of all, you have because obviously you're not going to get an RNA molecule that's going to know to code and to build the right components of a cell wall. So they come up with either two things. You either have it inside a little hole in a rock around one of these super vents, which churn out 150 amino acid proteins by the second, and ultimately they're in these little holes. So first, I have to get into the holes, whatever it is, this RNA thing, It's a tiny little hole, which is not, it's very difficult for things to get in and out of. So first of all, you've got to have a lot of material in there for this little RNA world, nascent RNA world, to start maturing and doing things. That's unlikely. You've got to remove waste products. That's unlikely. Although they'll come up with stuff, it's just nonsense, and it really is. And so, you know, but even if you get to that point that, okay, you've got something going here, you've got this little RNA world that somehow in one of these holes in the rock has chambers in a rock has suddenly started forming. How does it build a cell wall? It's protected, but one, there's no evolutionary advantage of building a cell wall.
There's no need.
If it did build a cell, it doesn't need. So there's no evolutionary drive. That's to be a drive. And if it did build a cell membrane or a cell wall, it wouldn't be able to get out. And if it did miraculously build all the right components, how would it code for it? And this is the point: how on earth would the code for a cell membrane appear in RNA? No one can solve, and they talk about, they're mixed together. No, the code, and this is probably one of the centers of the whole problem. The code in DNA, especially, is arbitrary and also abstract. So it's arbitrary in that you could reassign the code and it'd be fine. So you can take the code and you could change the code on. And in fact, you see this redundancy. You can change the code on, and you could move it to a different assignment and start again, everything will be fine. There's nothing intrinsic, or there's nothing about the qualities of the of the nucleic acids that form the code that determine the amino acids, right? So you could so a codon is one little piece of the code, right? It's 3 nucleic acids. It's 3 pieces of deoxyribonucleic acid lined up together. That forms the code for one amino acid that goes into a proton. Now, if you got a bucket full of that amino acid and you chucked this one mobile form of the codon, or you chucked a tRNA, which is the mobile form of the codon, into the bucket, it wouldn't bind to the amino acid. It doesn't find there's no physical connection. There's no physical meaning to the code. It doesn't. The code has to be translated, right? And the translation occurs in a cytoplasm using a protein, and these proteins are about 300 amino acids and more long. And on one end, you've got a bit that recognizes a portion of the tRNA, and at the other bit, you've got a bit that recognizes or it's all actually the same binding site. It brings the tRNA and the amino acid together, and then that goes back into the ribosome and gets bound onto the thing. So the translation occurs via a protein, and yet that protein had to be coded for. And there's never been any reasonable explanation as to how you get that system. No one has ever come up with anything that works, anything that makes logical sense as to how you get that system, because it's inconceivable how you get that system. You know, you talk about aptamers, but condensing an aptamer, it's a nonsense. They talk that, and this is why Tour was right when he said, for instance, there's thousands of scientists. Nonsense. There's very few in the field who are serious about this. There might be. So, for example, in our lab, when I was doing my PhD, because we dealt with nucleic acids and DNA and with amino acids, there was a chance we might do something by accident that shed light on these processes. And indeed, and I wasn't one of the people involved in this, but we knew something was something very exciting going on in the lab. And it's transpired that we had a publication in Nature that we discovered in one of our reactions a step in the prebiotic pathway that could lead to a solution, and it would have gone in Nature and it would have been a breakthrough. But it turned out as we were about a week away from publication. It turned out that actually it was that particular batch had been contaminated.
That's right. Yeah, that was in the early parts of your book, wasn't it? I remember you saying you were more of a bystander than a participant in that one.
Yeah, that's right. So what will happen is a lot of these publications will be byproducts of other work that's been done, and something will happen, and they'll do something. Oh, that. But very few people would make a career out of it. I mean, there is money in it. There's always universities, especially, and funding councils who are happy to bung a bit of money at this. But the reality is, if you can't conceive of how it can be done, you can't even come up with experiments to show how that might happen. So you can't conceive of how you would get this translation system, and it's abstract as well. So it's arbitrary, and for the fact the code it's abstract because sorry, you're nodding off, mate.
No, I'm with you. I'm just trying to kind of. It's just a lot of it is very much. It's just far beyond my knowledge.
Yeah, and it is most people, and it's kind of like, trying to help people to understand. I do try my book to do it really slowly, cognizant of the fact that this stuff's been, battering around my head for donkey's years. So I'm kind of familiar with it. And even then, especially on the chemistry, I'm not, I can't do it anymore. I don't follow it anymore. I'm just not interested. I've lost interest because I just know that you get someone like Farina stand up there and shout a load of garbage and look, oh yeah, look, good looking guy, young, cool guy, he must be right. And it's bullshit. It's just bullshit. It really, really gets on my tits, if I'm honest. But the fact is that, when I wrote my book, I've had very good scientists read my book. And they said, yeah, nothing wrong with that. I'm trying to think of something that's a little bit more.
Or maybe could you kind of expand on why purity is such an essential aspect of amino acids and what that means and what that would mean if it's not pure, and
I think if my thalidomide example doesn't explain it, then I don't know what will. You just get one. So it's just in one woman's body. This drug was given, so it's just one. I don't know exactly what it did. But probably inhibited some enzyme or did something. You just change one orientation and get that wrong, and basically babies come out with arms and legs.
So when you talk about purity, you're talking about chirality.
Well, but then you could. That's an advanced form of purity. So what you've got is reaction sites with a lot of these. So, for example, ribose ring has got, like I said, I think 5 reaction sites in its raw form, and so you have to make a nucleic acid, a ribonucleic acid. You have to put the base, which is there's 4 bases. There's 5 in total. You've got 4 for each of the RNA and DNA, and you swap the T and the U, and then you've got to join the chain with another. RNA. So, but there's 5 reaction sites, and basically they could react in different ways, and you get a mess. Now, also, if you have impure materials, there would be reactive sites in these impure materials that would then cross-react with your pure materials, and you just end up with a mess. I mean, when I did reactions in the lab, and it wasn't pure to start off with, or I got something ever so slightly wrong, even though I had all the instructions there and I tried to follow them really well, I'd end up with a brown gunk, right? It's the organic chemist's, nightmare, because it's impossible to get it out of the flask afterwards. But you get a brown gunk that doesn't have anything in it. It might have a trace of the element you're after. So purity is vital for getting reasonable yields out of reactions. And that's a key thing. The other thing that's not mentioned much is energy as well. That you need energy for some of these reactions, so you have to have boilings. But energy does 2 things: it can make things come together, that can break them apart again. Anyway, but purity is essential to get the kind of molecules that we're talking about. To make any chemical reaction work, you need good purity.
And that would be very difficult to come by on a proto-Earth, I suppose, environment.
Yeah.
Yeah. So again, just coming back to very big numbers in terms of, I mean, as you say, to conceive of 10 to the power of 168, as you say, that's a number double the number of atoms in the universe. And even,
No, it's not double.
Is it not double?
It's not double.
Oh no, it's not because it's. Going back to maths here, I can't remember that far back.
Yeah, so let's say it's 1 times 10 to the 80 is a number of. If you multiply that by 10, so you've got 10 universes. That's 1 times 10 to the 81.
That's right. Yeah.
So you've not doubled. You've multiplied
you're going by magnitudes.
Multiply it by 100, you get 1 to the 82. You multiply it by 1,000, so you start to get an idea. So it's not double. Universe times a universe. Unless you're good at maths, or you've got any. Unless you're putting numbers.
My math stopped at secondary school. Unfortunately, I didn't have much love for it.
Most people do. You get these guys, and it's fine that they do that. But is it? We've spoken about the other side of things. I'm much more interested in now. It's a lot easier to talk about. Frankly, people understand it. When I talk about NDEs, people get that, right? I talk about this stuff, people just sort of glaze over, and I get a bit, little bits that because I'm rusty. The ribose ring and stuff.
I think you really need that base, at least, kind of undergraduate level of chemistry knowledge to really begin to.
I did a PhD.
Exactly. So people like me have no chance.
Yeah. Well, it's even though my knowledge is ropey, my understanding, particularly of the origin of life, because I did go through a lot of it a few years ago, and then the reason I did it, the first time I had this real crisis of faith a number of years ago. And one of the things I'd always kind of believed, even during my PhD, was, these systems they're too complex to have come into being by pure random processes. I know chemistry, and I know physical chemistry, and I knew these things really well at that time, and it just didn't make any sense to me. I couldn't see how people around me. Well, actually, I'd say probably half my lab felt the same as I did, right? But they're less vocal about it. That's the difference. I talked to them, they said, yeah, they just don't think about it or not interested in it. And so many people I've spoken to over the years are the same, who have an understanding of this. And so, yeah, you get people like James Tour who the same, they just get it. But I at that time, I was really struggling, and I had some time. So I just went, it's like 2008 I did this. I just went to the original literature and everything on the origin of life research. And honestly, there isn't anything really much more illuminating since then. 10 years later, Sutherland wrote his paper, and then, go another 7 or 8 years. Nothing really major has happened, especially in terms of solving the conceptual issues. And I just really dug deep for myself more than anything else. And I just realized that it's impossible. The first thing I wanted to prove to myself or understand was this chicken and egg paradox. And I've read so many times by scientists that no, RNA can translate, DNA is translated in the ribosome by RNA. It's not. It's actually the translation occurs in the cytoplasm with these 20 amino acyl RNA synthetases. All that happens in the ribosome is assembly of the proteins along the mRNA template using a tRNA that's already got the amino acid. Again, I'm starting to go off. But so I just really looked into it. And then about 7 years later, I was in a church and I was speaking to someone, and they said, "Oh, you're a scientist. Why do you believe that, what did you believe that, in evolution stuff?" I said, "Well, evolution, I'm not really an expert on. I understand that there are problems with the emergence of new phenotypes and new species. But I worked in HIV for 13 years, and so I've seen evolution occur. Yeah. However, there's a limitation to evolution that's very apparent, even with DNA, that you can only make very, 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 very minor modifications, but everything goes wrong. So you make most mutations are either harmful or don't do anything at all. Very few mutations in the code are beneficial. And in one cell cycle, you'll get hundreds of mutations, maybe thousands in one DNA in HIV. But most of those will be innocuous. They won't do anything. polymorphisms, and it won't affect the function of any of the proteins that are being coded for. Every now and then you'll get one that changes the function, and it'll usually make it worse. Every now and then it'll be better. But to get two in one cell cycle change, it happens, but it's quite difficult. It takes longer. And all the evidence for this is really clear in how when they first thought there was a massive breakthrough in RNA with HIV with AZT, people started developing resistance within a few months. Then they added another molecule, I think it's DDI, I can't remember what it was now. There is something else in that, had a slightly different focus in terms of its impact, and it stretched out things for a few months instead of a few weeks. When you got to 3 drugs that targeted 3 different sites within either 1 protein or 2 proteins, then you stopped HIV production. Because even though you might have, I think it works out, about 5 billion, it's about 5 billion, in someone who's got quite a high viral load, which is quite common, especially in those days, you might have 5 billion HIV viral particles in someone's body. You've got about 5 liters of blood, and you can get about 1 million HIV particles in 1 milliliter of blood. And someone who's really highly infected, right? And if you give that person 3 drugs that target 3 different things, the virus just won't completely disappears. It just goes into hibernation where the drugs can't get. That's why you can't cure HIV, because if you remove the drugs, it comes back up. And so what has happened is, out of those 5 billion HIV virions, not a single one had an HIV virion that was able to, that had 3 mutations in it that were successfully able to fight 3 different drugs, 3 different things. And then you've got millions of patients. So you've got millions of 5 billions who have been treated with these combinations successfully. And, so that shows you how difficult evolution is. It's extraordinary. And HIV is not particularly well regulated. It can function under fairly, humans are much more, we have regulatory processes that, make sure that none of the rubbish gets through. But it's really difficult for big changes to occur.
Although, is it not, again, the argument that over millions and millions and millions of years, that then becomes a lot less kind of absurd to think about?
I think, there's an argument for that, which is why I don't tend to get involved. If you've got the starting system, if you've got a cell with a 20 amino acid, DNA-based code and translation system, you've got that to start with. I struggle with it, but I could say, yeah, it's feasible that over 3.7 billion years, you go from that to us. I think, Michael Behe, I think it's no time pronounce his name, he's made a lot out of intelligent design, and he uses a lot of these arguments, and I struggle to disagree with him, actually, because even in evolutionary biology, there's questions now around how you would get a new phenotype appear, even over a very, very long period of time. So the problem with a long period of time is that one of the laws of evolution, whether it be chemical evolution or whether it be biological evolution, whether it be pre-Luca evolution or post-Luca evolution, is that any change has to be beneficial, and it therefore has to outcompete the other molecules, so predominantly. Otherwise, you've got millions of other cells being produced, and if there's nothing special about it, it'll just disappear. Now, bearing in mind what I just said about how difficult it is to get a successful alteration, then, it's conceivable, or it becomes inconceivable that through one mutation, you're going to suddenly get a nose. You might get a slight nose or something. I can see, I got to see in Richard Dawkins' arc, and I kind of think, he's more of an expert in evolutionary biology than I am, so I'll give him that. But it just becomes a little bit, strange. But when you apply it at the chemical level on the areas I'm talking about, it becomes absolute, these changes. And if you don't see, you'd have to, like, for instance, to go from you can't, so using your example, you could have an evolution over millions of years or whatever. You've got 100 million years, right? So let's take another example I gave you in that little slide deck that I'm working on. I'm going to put out there. And this is this is the last step to Luca, right? The last step to Luca. So we have a 20 amino acid system. That's what our proteins are made of. There are a few examples of, species that only have 19. I don't believe there are examples of previous, and there's no one actually believes that there are examples of predecessors. They just don't happen to need the extra thing. And there are examples of other things, but they're a bit of a distraction. They don't really help, which is why not much is made of them. But let's take the 20 amino acid system that we've currently got and the DNA code that codes for it, right? Francis Crick, who was one of the co-discoverers of DNA and then who went through the process of finding out what the code meant. Well, after they completed that process and they discovered this amazing relationship between the codons and the amino acids, he concluded that this happened once in nature and then froze. And the reason he gave for that is because this system is so complex that you change just one thing and it falls apart, right? There are all these interdependent things. So, and again, I'll start getting into the weeds of this, but and he's right. But so's a 19 amino acid system. There's no difference really in terms of the complexity, apart from one amino acyl RNA synthetase. That's the only one of the key differences between that and the other. But you'd have to have one of those made. But you'd also, and this is where it starts to get really, really where you start to think, yeah, this doesn't make sense. You've got a 19 amino acid system that's presumably functioning quite well. Why would it suddenly start coding for another amino acid? Where did the amino acid come from? Where do the proteins that make the amino acid come from? How does it know where to put the amino acid in the proteins it's now making? Everything falls apart. But all of that has to happen in one cell cycle. Otherwise, there's no benefit. You can't have saying, it produces an amino acid and it just floats around for a million years and doesn't do that. The amino acid will get in the way, react with other stuff, and it would have no meaning. You have to have the code for it at the same time as you have the new codon, at the same time that you have the new protein that binds the tRNA to the amino acid, at the same time that you have the new protein that appears and incorporates it. It's beyond insanity, which is why no one can explain it. It's inconceivable. It's actually inconceivable how you overcome this problem. And that's just one little step. Going from a 2 amino acid system to a doublet system to a triplet system is like a whole different magnitude of insanity. Going from a ribonucleic acid system to a deoxyribonucleic acid system, another whole level of bonkersness. Getting the code in there in the first place, bonkers. It's all completely bonkers. To someone who understands this, if you're really thinking about it, it doesn't make sense. I'm not sure how long these people have actually thought about it, because I don't see how someone who's intelligent and has done the science and really got to grips with it cannot see how that is inconceivable. The only way they can see it is that they have greater faith in science than I have in God, right? Because it's scientism. To believe that you can find a random natural path to doing that is scientism. It's an unrealistic faith in science. It has no basis in fact or evidence. It's complete fecking nonsense.
Or at least promissory at best,
What's that?
Promissory. Is it promissory? Where, we haven't found it, but we will eventually find the answer to this at some point in the future.
Yeah, it's faith. It's faith in science. It's not faith. It's not grounded in reality. The fact is, we have known about this system and how it operates since I think it was the early to mid 60s. They elucidated the full code. I can't remember exactly when it was, and how it all operated and how it was translated. So we've known for, I think, 60 years how it all works. And you we can't even think of, Francis Crick's frozen accident is just one problem, and no one can answer it. His solution was panspermia, which is shipping it in from outer space, but it just moves the same problem somewhere else. And that doesn't help.
Yeah, because you've still got then the problem of, well, whether it's Earth or not, it's the same issue. Yeah.
So I don't know. It's but yeah, it's scientism is something which we see all the time.
And it's quite interesting to see the parallel that over the 60 years, such little progress has been made to answer that hard question, and yet it's accepted as kind of the most likely. And yet something like psi, which has equally kind of not had much progression in the same kind of length of time, that is used as the excuse as to why it must be nonsense, because it hasn't progressed in however long, even though it has.
Well, what do you mean? So, what you said, psi?
Psi, like psi phenomena,
right?
Parapsychology. Because one of the major arguments I hear about parapsychology is that it hasn't made any advancement in our knowledge of the phenomena, so therefore it's bunk. Whereas equally, you could say that about origin of life research, and yet that's considered the...
Which is why they'll, put up these papers, because, the truth is that the academic establishment has a bias towards atheism. It is left-wing, right, which is atheist in nature. It has to be by its very nature. And it is ultimately atheist. That the academic establishment it's atheist in its politics, in the way that it looks at the world, in everything. And it's also science itself. It works from a founding principle of atheism, but that's not scientific to do that. To discount an alternative explanation, it's called methodological materialism. It's basically an unwritten mantra, but there's always an answer that can be answered by science. Well, maybe there isn't. Maybe
I think, in a way, it's very much misrepresenting what science is, which is adapting to alternative explanations rather than assume. Because you assume the scientific method is ontologically accurate, if that's right, for everything in the universe. That's, the very scientific method is assumed, which is quite paradoxical, really. But
it is reliable if you
apply
it without bias. If you apply it without bias. So, in my book, I go through this, and, there is really no real evidence that the origin of life has a random, natural, there's no evidence really at all that it could occur by random natural processes. That's just a fact. There really is no evidence. There's huge mountains of evidence against it. And I show that in my book, and I've sort of slightly covered some of that here. These conceptual problems are too big for people to conceive of how you might overcome them, let alone prove that they were overcome that way. So there's a lot of evidence against it. But if that was the only hypothesis that existed, if that was the only possible explanation, you still have to go with it, right? Because, there's another explanation. And that is how atheists think. They think that's the only explanation, so there has to be an explanation. So there is an explanation. So there is an explanation. There isn't,
and it will be found.
It will be found. We found that. And don't look too closely, but yes, it is. It's bollocks. But ultimately, that's what they do. However, there is actually an alternative hypothesis when it comes to the origin of life, and I'm not talking about intelligent design. Intelligent design is a qualitative thing. Whereas intelligent initiation is just about the origin of life. It's just about this system in particular. And it's just really about the DNA code. And the fact is that there are millions of codes in existence. Right? There's only one, the DNA code, that we are aware of that we can't show with absolute certainty it had an intelligent origin. That is scientific evidence, albeit, whether you call it circumstantial or whatever, it's not descriptive. It's real evidence because it is a code. It's abstract, it's arbitrary. It's, the idea that you've got this chemical structure for a completely different chemical system, that they don't speak to each other directly, and that in this one chemical system, which has its own three-dimensional structure, codes for a completely different three-dimensional structure in a different chemical system. It's insane. Anyway, so I'll start going off again. But
so yeah, point being, when you think about intelligent design, you're not thinking Adam and Eve and all the species at once. You're talking about some form of intelligence behind the initiation of Luca, I suppose.
Yeah, well, and specifically the DNA code. For me, it's, you know, Luca's the last universal common ancestor, but it's specifically the DNA code and the translation machinery and system around how it then produces these three-dimensional structures that are so far removed from the structure and, you know, of the code itself, the non-abstract, the non aspect of it, that, and there's no way of conceiving how that happened. And people have come up with stuff that frankly is garbage. And I talk about aptamers in my book, and it's just, I call it, because the RNA world is a red herring, it doesn't help us at all. It doesn't get us to DNA. It doesn't get us to a DNA-based world at all, because you've got to get rid of one of those oxygens. And you have to do it in one go. Happy days trying that. And then you've got the, you know, lost my track. Anyway, there you go.
Interesting. So I suppose kind of the reason that it's assumed that the atheistic non-supernatural, for want of a better word, explanation is likely to be the case is because it historically always seemed to have been the case. You know, with historic discoveries through things like lightning, like you know, various phenomena that we've discovered were previously.
Science has obviously shown that a lot of things that people believed was nonsense, right? And that's true. And it's, you know, it's pretty much solved or shown us that most of those things are the case. But what's different about this is that if you believe in God, then you believe that he did make everything, right? Now, again, I go back to what I talked about when I spoke to you, I think the last time or the time, I can't remember what it was I spoke to you about it, but how, you know, really this life isn't real. It's an illusion. That Christians don't like hearing that because they think it's real. But I believe that it's an illusion created in which we live for a purpose. And I don't want to get into all of that now. And it's created by God. I'm not talking about philosophical idealism or anything like that, but it's you look at the quantum mechanics and how things only become real when they're observed. I don't understand why that is, but I'm told it is, and I accept it. And Einstein and the other geezer, what was that?
Planck?
No, I think it's Planck. It was Bohr, I think. Some Swedish guy. It wasn't Planck. They argued about this, about whether what we see is real until it's observed. And Einstein lost. Right? And the other guy, Niels Bohr, I think it is. And they argued about this, and it ultimately, quantum nature of things is such that the only reality is experience. And so you could argue, and here I'm going off on one, and I'm not saying this is the case, but, you know, all this origin of life stuff, it's just a distraction, you know. If nothing actually exists in reality, then it's just it's just a puzzle put there to distract people. And if you don't want to believe in God, feel free. It gives us free choice. You know, you can believe in scientism if you want. You can believe in scientism. But for those who believe in God, there is enough material in this to say, well, it couldn't have happened by random natural processes. And we know that God is obviously intelligent if he exists, and that there's evidence for intelligence. And so there's enough here for those who want to see, just like there is with NDEs. There's enough there for people who want to see, just like with so many aspects of the faith. But for those who don't want to see, there's enough for them not to see. That's exactly how it's supposed to be.